Okay, once the polypeptide chain is made, we call it a primary structure. So it's basically the order of amino acid. After finishing this long chain polypeptide, the exact order of the amino acid is called primary structure. Then after that, the amino acids with each other, they can make different bonds because their R groups are different. Their R groups are different. These R groups can be found in many different variations. Some are polar, some are non-polar, some has a charge, some hydrophobic, like many properties owing to this R group. As a result, between amino acid, they can interact with each other. So secondary one, amino acid make a hydrogen bond. Here it's only the hydrogen bond. So they make hydrogen bond. Then we can identify very special shape. So this shape is sometimes like this spiral helix. The polypeptide chain get this kind of a shape, and the hydrogen bond, the red color ones are the hydrogen bonds. They can create the shape. So the red color one is a hydrogen bond. And there's another form available that's they're making this kind of a sheet-like appearance. So again, the hydrogen bonds make this shape. And this is called alpha helix, and this is called beta pleated sheet. So example is we can observe very nicely the structure. A protein called keratin. It's on our skin, the layer and the hair. They have this keratin protein, and then silk. We can find this structure. Now in tertiary form, the polypeptide chain can make a lot of foldings. So here only we observe only two shapes, but then the tertiary structure, it's making so much of foldings like this. So when they make the foldings, finally it gets the structure like this, like a 3D shape. Now it's create the final version of proteins with the 3D shapes. And here between R groups, let's say one amino acid of R group and then another amino acid of R group, they can make bonds. So here again another bond. So here another bond. And here another bond. So they can make a lot of bonds, and the bonds are four types. One is hydrogen, other one is a hydrophobic. If there's a lot of carbon and hydrogen chains of this R group, they can make a hydrophobic bond. And some amino acid has sulfur group, they can make a disulfide bond. And some can make ionic bonds. Here ionic bonds, disulfide bonds, hydrophobic bonds, and hydrogen bonds. So we can find 4 types of bonds between R groups, and it creates a 3D structure like this. So we can see most of the proteins, they exist in this 3D shapes. You will see later on, end of the lesson, if chemicals which can break these bonds, they completely change the shape of the protein molecule. If the protein molecule shape the shape change, its function lost. So it's not properly functioning. And the quaternary structure defined as combination of several tertiary structure molecules. So once it get the tertiary structure and several tertiary structure proteins join together, and they can also make bonds, especially they make the hydrogen bonds and hydrophobic bonds. So they can create this a big structure. Example is hemoglobin. Hemoglobin has a quaternary structure. So that's how the 4 levels, primary, secondary, tertiary, and quaternary structure existing in proteins. Alright, so if you understand well the level of proteins, primary, second, tertiary, quaternary, we can move to the next, to the denaturations, right? The topic called denaturation. So we will see what is denaturation. But before that, I am again telling, if chemicals can break these bonds between R groups, they lost the shape. If they lost the shape, it can't reform, because the folding is done inside the cells with a very special mechanism. When the proteins are made within the cells, they use these special mechanisms to create the shape. So if chemicals add to the proteins, they can break this bond and it completely disrupt the structure of the proteins. So this is called denaturation. So let's see what kind of a chemicals, what kind of a method we can destroy the proteins, denaturation of the proteins, right? Okay, I hope you have copied this. Let's do this question. This is again exam question. The chemical formula given above represent the basic constituent unit of, it's a straightforward now. Yes, I got several answers. Yes, this is the amino acid. You can see a amine group, carboxylic group, and hydrogen. And this part is same in all amino acid, only the difference is this R group. So amino acid join together to make proteins. Lipids, basically glycerol and fatty acid. Nucleotide make the nucleic acid. Alpha glucose make the starch. Beta glucose make the cellulose. Right, there's a question, what is the difference between secondary structure and the uh, tertiary structure? So I can show you the way people construct the shape of the protein molecules. And if we look at these protein molecules, when we identify this helix, and this part represent secondary structure. So within a protein molecules, we can find a special shape like this, then we call it secondary structure of the proteins. So they make a long helix, that is a secondary structure. But the tertiary structure, the protein polypeptide chain folding to make the 3D shape like this. So it has so much of foldings to make a 3D shape. So then this helix represents secondary structure of proteins, and these kind of quite huge foldings represent tertiary structure of proteins. So when protein exists in these shapes, we give a name. This is secondary structure, this is tertiary structure. Alright, now we come into the last part, denaturation. Denaturation of proteins. Right, we'll take a common practice we did recently, washing hands with sanitizers, washing hand with alcohol spray to the hands, or maybe washing hand with soap. During COVID-19, it is practiced across the world. A lot of people start to wash their hands with soap and water. If not, they use the sanitizers, spray alcohol, 70% alcohol, and wipe the hands with alcohol. Why they did this? What is the reason? How it help? Do you know what is the science behind this practice? Because the general public usually complain, if we can destroy COVID-19 with just soap, why we cannot make a method to completely remove the virus? Right. The reason is this. Now, if you look at the structure of the virus, they have a nucleic acid. This nucleic acid is DNA or RNA, And virus, COVID-19 virus, they had a RNA nucleic acid. And this nucleic acid is covered with a protein layer. This protein layer is called capsid. And on the top of protein layer, there is also a lipid envelope, and we can also see attachment proteins like this. Now this attachment protein shape is exactly fitting with human cells, because human cell has another protein which is exactly fit to this protein. Let's say this is a human cell. So this is the reason the virus can infect human, because of this protein on the virus can attach to human cells, because human cells has the exact shape which is fit into this protein. So when we use soap, when we use ethanol, as I mentioned today, these are proteins. So what will happen, these chemicals can break the bonds, and they change the shape of the proteins. Let's say if the soap and alcohol is used, this protein is shaped the change. Now this is not fitting to human cell now. So it destroys simply the protein on the surface of the virus by the process of denaturation. Right. What are the other agents which can denature the proteins, destroy the proteins? So we list all this. Here is the list. High temperature and high energy radiation. So if we fry egg, the egg protein is albumin protein, egg white is albumin, and the protein is destroyed. Then strong acids, bases, high concentration of salts, heavy metals, detergents, urea and ethanol. So this is the list which can destroy the protein structures. Right, this is another question from the recent exam. Can you try to find the answer for this? Yes, if we correctly understand the lessons, the secondary structure has a hydrogen bond. Disulfide bonds we can find in the tertiary structure. And 3D forms, we have disulfide bond. And it is not 26, it's 20 amino acid. Carbon, hydrogen, oxygen, nitrogen, sulfur, not phosphorus. And here we learn detergent can still destroy the proteins. And we learned previously, remember the hamster? T is for transportations. So protein is important for the transport of material within the blood and also in and out of the cells. So protein is a good transport molecule. The answer is this one. Right, do you have any questions? If not, we can come into the last lesson, the nucleic acid. Right, we come to the nucleic acid, DNA and RNA. Deoxyribonucleic acid, and the other one is ribonucleic acid. So these both molecules, DNA and RNA, the monomer is nucleotide. Let's first look what is the monomer of DNA. This is the ribose sugar, and ribose sugar has 5 carbons, 1, 2, 3, 4 and 5. And one N joined with a nitrogenous base and the other N joined with a phosphate group. And when we numbering the ribose sugar, we are giving a small dash like this called 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So we use the word prime. It's different to glucose-like molecules. We don't use prime as a word. We use 1, 2, 3, 4, 5, 6 like this. So it indicates the prime word is given to the DNA and RNA molecules because they are precious molecules. They are very important molecules, right? And this is called a nucleotide. And when you're talking about DNA, we call deoxy. That means second carbon position, the oxygen is lost, it has only hydrogen. When you take RNA, it has O here, OH group. So if I draw this has OH and nitrogenous base and the phosphate group. So this belongs to RNA, this belongs to DNA. That's a ribose. This is a ribose sugar, this is deoxyribose. You see the difference. Here there is a OH group and here H. Do we need to memorize the structure of carbohydrate, lipid and protein? Well, remember the MCQ we did. Amino acid is given, you have to identify. Glycerol is given, you have to identify. So the two examples indicate at least important structures like amino acid, glycerol and glucose molecule. Better to these molecules, the basic structure, memorizing may be beneficial, because we have seen MCQs. You need to identify glycerol molecule, you need to identify amino acid. So basic molecular shape, better to memorize, not detail bond formation and then the memorizing the different bonds like things you can leave at the moment. Okay, these are the monomers called nucleotide, and the difference is DNA has deoxyribose sugar, RNA has ribose sugar. Right, now this nitrogenous base here, there are 4 different nitrogenous bases. So these are adenine, guanine, cytosine and thymine. So we abbreviated this capital A, capital G, capital C, capital T. Adenine, guanine, cytosine, thymine. And RNA, they also has 4 different nitrogenous base. So they are adenine, guanine, cytosine. Instead of thymine, it has uracil, abbreviated by U. 1, 2, 3, 4. 1, 2, 3, 4. So DNA has 4 different nucleotide. RNA also has 4 different nucleotide. But this nucleotide, only small difference. Thymine is in DNA molecule, uracil is in RNA molecule. Okay, again we place these nitrogenous bases into 2 groups, because the one type of nitrogenous base, they have 2 rings like this. The other type of nitrogenous base, they have only 1 ring. So this, you can remember, this has a few rings. As a result, it gives the name purines. This is called purine and this is called pyrimidine. So purines are adenine and guanine. Pyrimidine are cytosine, thymine and uracil. These informations are very important, not like a carbohydrate or fat or other things. DNA molecule again and again we have to learn, because molecular biology we have to learn again DNA. So basic information of DNA molecule, like these are the basic information. It is better to remember this information. Right. Now if you copy this one, where we can find the nucleic acid in the cell, we're going to answer the question, because proteins, carbohydrate and lipid we have seen they are in many places, cell membrane and the structural component of cell walls, and they make the cell membranes and other part of the organelles. So where is this DNA or RNA molecule we can find in the cells? So to answer that question, we try to answer this question, the MCQ questions, right? So this MCQ question, even though you haven't learned subcellular structures like organelles, in eukaryotic cells, in addition to nucleus, DNA is present in, okay, eukaryotes are nucleus containing cells. Prokaryotes are bacteria which do not have a nucleus. So where do we find DNA in addition to nucleus? Probably too early for you to answer this question, but we want to know at this place the location of the DNA molecule. The idea is not to answer very well this question. Once you learn organelles, you can answer, but we use this question to learn something. So I'm getting some mixed answers now, because I know that it is too early to give a good answer. It's fine. But we want to learn something, right? It's not just, we are not going to find the perfect answer. Yes, I see some mixed answers. Okay, we can find the DNA molecule in a cell, both mitochondria and chloroplast. In addition to nucleus, we can find in mitochondria and chloroplast. The answer is 2. It is not only the nucleus, it is also found in mitochondria and chloroplast. We will see what is the role of this DNA in mitochondria and chloroplast, and why mitochondria and chloroplast have DNA. But for the learning purpose, DNA can be found in mitochondria and chloroplast and nucleus, right? Okay, so let's move to the next level. How this DNA molecule can be formed. So that is lesson on the join nucleotide to make single strand, and they draw double strand, right? So let's come into the DNA molecule, the formation of DNA molecule. Here is a one nucleotide, and the front side of this one is 5 carbon, so 5 prime carbon, and this side is a 3 prime carbon. So 5 prime carbon and 3 prime carbon location is this. Right. I think we are reaching to the end of the session, so we can join after the session to continue the story. So if we take another nucleotide, this nucleotide attached like this way. Now here 5 prime end and here again 3 prime end. So it create a bond between this carbon and phosphate group like this. So now all the nucleotide can line up like this way to make a long chain. So I draw only here 4 nucleotide. So 4 nucleotide join, and it's still, this end is 3 prime end. The front one is a 5 prime end. And this phosphate bonds is called phosphodiester bond or backbone. Phosphodiester bond create the phosphate